Hello and welcome to this webinar titled Strategies to Overcome Low Endotoxin Recovery Using the Conventional LAL Assay, hosted by Biopharma Asia magazine and presented by Dr. Ruth Daniels, Senior Scientist, Microbiology Expert at Janssen, and Kevin L. Williams, Senior Scientist, Endotoxin at BIOMÉRIEUX. My name is Orsolya Balog and I will be the moderator. Now, please allow me to introduce our first presenter, Dr. Ruth Daniels, Senior Scientist, Microbiology Expert at Janssen. Ruth Daniels holds an MSc and PhD degree in Bioscience Engineering from the Catholic University of Leuven, Belgium. After an academic career, she started in 2011 at Janssen, Sanofi, Belgium, as lead microbiology, operational, technical support, and manufacturing science. Since 2015, Ruth is the senior scientist at Janssen within the Analytical Development Microbiology department. She has a broad expertise in the field of endotoxin and pyrogen testing and is involved in multiple Janssen clinical development programs as microbiology expert. Ruth is a member of BPOG Low Endotoxin Recovery Group and PDA Low Endotoxin Recovery Task Force. Welcome, Ruth.
Thank you. I would like to thank you for the opportunity to present here our study, our data, and our approach to overcome low endotoxin recovery using the conventional LAL assay. So my name is Ruth Daniels. I work at Janssen, that is part of Johnson & Johnson. I live and work in Belgium, but we collaborate a lot with the people around the world because I'm part of the Analytical Development Microbiology Group, and that group is responsible for all the new pharmaceutical products that are produced and or in the pipeline. All new clinical molecules needs to be tested and it needs to be screened for pyrogens. And then a few years ago, all of a sudden, there was an issue with the low endotoxin recovery, so with the fact that endotoxins were lost or were not detected in certain matrices. So now we developed we globally implemented our strategies to overcome low endotoxin recovery for the pharmaceutical products intended for parenteral use. We do we do a training of the people around the world. We do a global rollout of all the things that we've learned, and we do global method transfer after the development and the qualification. So the conventional LAL method is a method that detects pyrogens that are produced by gram-negative bacteria. There are other low pyrogens that can be present in your pharmaceutical product, and those can be detected by the rabbit pyrogen test. That is a test that is able to detect all pyrogenic substances. But most of the pyrogenic substances that are in our products are pyrogens from gram-negative bacteria, and therefore the BET is the most commonly used test to screen your commercial product and your clinical products. Low endotoxin recovery in a nutshell. There can be two different types of low endotoxin recovery. One issue is when there is an interference with your working with your LAL. So that is your enzyme that you need for your detection of your endotoxin. When there is a pH effect, your enzyme will not work properly, and then you will not detect the endotoxins that are present in your sample. Whether there are chelating agents available, whether there are protease inhibitors available, they can all interfere with your LAL enzyme. On the other hand, there can also be endotoxin interference because your endotoxin can absorb to the container or can absorb at the protein. And then you can neither find your endotoxin back. Or there can be aggregation. When you have chelating agents and surfactants in your product, then aggregation can take place, and then you have also a problem in the detection of your endotoxins. Low endotoxin recovery, in our hands, we have seen that magnesium is very important in this whole story of detecting or non-detecting your endotoxin. Magnesium sulfate is a cofactor in the reaction of your during the detection of your endotoxin, and magnesium sulfate is a part of the biologically active LPS aggregates. And that is what you see here. When there is, for instance, citrate available, present in your sample, the citrate can remove your divalent cations from LPS aggregates, and so-called fragile LPS aggregates can be formed. When detergents are present on top of that, they can remove the LPS molecule from the fragile LPS aggregates. So magnesium is a very important player. The strategies now to overcome LER using conventional LEL methods, well, in this presentation today, this presentation is divided in three different parts. First is the endotoxin hold time studies to identify low endotoxin recovery. In the second part, I discuss with you our in silico assessments of the interfering factors and the associated low endotoxin recovery mitigation strategies. And the third part is a case study that from the shelf that I can share with you, where we optimized where we did an optimization of the product-specific LAL assay to overcome low endotoxin recovery. First of all, endotoxin hold time studies. We perform the BET during our process validation to ensure that there are no paragenic contamination present. We test our commercial batches routinely, and we do a requalification of our BET when there are process or site changes. We do low endotoxin recovery studies for our drug product, and in the endotoxin hold time studies, we do that on the profile batches, and those data are submitted in the BLA or in the MAA. And we use a harmonized endotoxin recovery study protocol that is published by the BIPOC group, by Bolden et al. In 2017. When we perform endotoxin hold time studies and we assess problems, well, then is the next step that we do an in silico assessment, we search in the literature to see if we can find solutions, mitigation strategies to overcome the problems. And that is the next part that I would like to share with you. And then we do an optimization of our product-specific LAL to overcome our LER. Well, this is now the second part of the presentation, and here I would like to show you how it works. This is a summary of the literature that was, so what I performed is a very robust and huge literature search. There is a lot of literature available on the market. There are books available, and there is a lot of good information available. What I did is then I made a table, and this table, this in silico assessment, is used in all our low endotoxin recovery mitigation, because for each of the interfering factors that are described here, first is is a pH problem. There is then an example from the literature given that can be an acid or a base. An example that is relevant for the pharmaceutical product can be the pH of your drug product. You have to take into account the pH of your drug product. And what we do then when we have an issue is to screen our molecule or matrix and assess whether or not each of the interfering factors that are shown here, it is the pH, but also divalent cations can be present, or chelating agents can be present. Well, they can cause problems. And for each of the problems, each of the interfering factors, here is a summary of the mitigation strategies that are available in the literature. And I will not go step by step to all the mitigation strategies, but I will now, with you, just highlight what are the examples, typical examples from the literature and typical examples from the pharmaceutical products that we face. So if we have, I just discussed with you that it can be an acid or a base. It's a second type of interfering factor can be calcium. If there is too much, if there are insufficient divalent cations, it can be a problem. Third problem is type of interfering factors or chelating agents. Examples from literature from chelating agents or citric acid, sodium citrate, or EDTA. And examples in our pharmaceutical products that we typically work with, or L-histidine, or PS80, so polysorbate 80, or sorbitol. So this is applied to our products. This is typically from literature. And in the last column, but I will not go to all the samples or solutions or mitigation strategies you can have a look at when you go through the presentation. And when you have this, by yourself a complex sample, and when it contains, for instance, chelating agents, then you can have a look at the list of the mitigation strategies, and then you can start your development work to try to solve the problem. Surfactants is another type of interfering factor. An example that we typically see is PS80 or sorbitol. proteins and protein contaminants or monoclonal antibodies can typically interfere with your endotoxin. Typically, it is very unpredictable, the interference of your protein, and it depends of the molecular weight of the PI of your product. Then there is another category of interfering factors that are agents that denature the protein, and the protein that is then the Lal enzyme. Agents that typically denature the Lal enzyme are agents that cause for high osmolarity, or heavy metals, or surfactants. And in our hands, in our products, examples are sorbitol, PS80, and here also sorbitol. So another class of interfering factors is the protease inhibitor. It can be, a typical example from literature is human plasma. Another interfering factor category is a container leachable. An example is polystyrene or polypropylene. We typically test in our final container. Then next to that are lipids and liposomes that can typically interact with your enzyme, with your endotoxin. Metal ions can cause problems. Metal ions in our hands can come from stainless steel or from the raw materials itself. Examples are aluminium, iron, or zinc containing formulations. The third part of this presentation today is the case study, because I would like to show you how we deal with the optimization of product-specific LAL assay to overcome LER. Well, we have, this is a real example. This is a sample, a pharmaceutical product, and it contains a monoclonal antibody. It contains the L-histidine, sorbitol, and PS80. So I just, if you recall correctly, then you know that I just mentioned those because they were examples in the list of the mitigation strategies and interfering factors a few slides ago. So those can, or known in literature, they can interfere because they can have chelating agents. If they have chelating properties, in the slides before, I just demonstrated that in that case, if you would like to do an optimization and do a development, you could try 4 different options to try to solve that problem. Well, that part of that matrix that we are now working with also has surfactant capacity. For that, there is one option that we can try. There is an agent in it that is able to denature the protein. And there is a monoclonal antibody in our sample, so 5 additional options, mitigation strategies that we can try during the development. Try to solve the problem, try to optimize the problem of low endotoxin recovery. There is also, there might be an issue with iron leakage from stainless steel. So that counts for another 3 options. So in phase 1, we do method development, and we start with one of all the options because we believe that the interference with the magnesium is very important, as I explained to you just a few minutes ago. In the second phase, we do, after the method development, we do a qualification of the optimized routine method in our final drug product container. And in the third phase, we do an endotoxin hold time study to verify the optimized method. Well, this is the buffer, the magnesium sulfate tris buffer. That is a cation exchange buffer that is available that you can buy and that you can use in your lab. And important is that this can work on the magnesium depletion, so that because you need your magnesium as a cofactor and because it is a part of your biologically active LPS aggregate. Important is to make a dilution range of this buffer, because you have to optimize the dilution for your dependent on the vendor of the lysate. You have to optimize your dilution. This is the development, the experimental setup of the development. We work with KCA, so kinetic chromogenic, and we have a 5-point standard curve. We spike at time point 0, and we spike in our undiluted DP in its final container. We always work with a positive control that is low endotoxin, Lal reagent water as a positive control, and we store everything after that it is spiked. And we measure things at time point 0, and after 4 weeks, we do a product dilution of 100, and instead of diluting the product in water, now we dilute the product in the buffer, the magnesium sulfate tris buffer, and we try a range between 1 and 10 and 1 and 500 dilution of that buffer. Important is that our positive control has a good spike recovery, so the spike in water, and that for a whole time study. Important is that for each time point, we compare the recovery of each time point with the recovery at time point 0 in our product. If the recovery is between 50 and 200%, then it's fine. And we always work with net spiked endotoxin concentrations. So that means that we, if there is endotoxin, if you measure endotoxin in your unspiked sample, we calculate the net spiked endotoxin concentration. This is the development data. Here you see so we spiked with 2.5 endotoxin units per ml. This is the amount of endotoxin that we measure after time point 0 after 4 weeks, and this is the range. This is the screening. That is a range of the buffer that we used to dilute our product in. Here you see the recovery, the PPC recovery, and a good PPC recovery is a PPC recovery between 50 and 200%. So the bold one is a bad one. So this is promising. But then this is our product that is spiked with an endotoxin. Next is a very important control to take into account. That is to take your unspiked drug product into account, because when you measure in routine, then you measure your unspiked drug product, and important is also to have a good spike recovery of your unspiked drug product. So we diluted our unspiked drug product in the same range of the buffer as a control, And we see, for instance, here, after 4 weeks, when we have recoveries higher than 200%, that is not okay. So here we are running to the extreme, to the border. We are very high in the PPC recovery. So, and that is not what we would like to have. So therefore, we decided that the 1:40 buffer is actually the best buffer dilution to dilute our drug product in. That is also here concluded in this slide. So we have demonstrated that a 1:100 dilution of our product in the 1:50 dilution of the magnesium sulfate tris buffer is successful to overcome LER. Next step is then a qualification of the routine test method, and the qualification of that test method is always performed on 3 batches, and a validation of the whole time study on 3 batches. And that is shown here. What we did then during the whole time study is exactly the same as what we did during the development, but we used 3 batches and we measured weekly. These are the results. Same table, same interpretation. We spiked with 2.5 endotoxin units per ml, and these are the results after 1 week, after 2 weeks, 3 and 4 weeks. And here is the PPC recovery. This is the positive control that is included in the plate. And on the next slide is the recovery of the endotoxin that is spiked in our undiluted product, the recovery of that endotoxin in the 3 different batches over time. And for that, I repeat it is very important that you compare your recovery of your product at each time point with the recovery of your product at time point 0. And that is shown here. So that is a very nice recovery. So with that, we can conclude that our magnesium sulfate tris buffer that we used for this sample to solve the issues here is useful. We used it in during the development, but after that development, we were able to validate the method, and we were able to perform a whole time study, and we were successful in the recovery of our endotoxins over a 4-week whole time study. And then, last but not least, I would like to thank my co-workers. This is not a work that I did on my own, but these are the people in the lab and people, collaborators in the US that are responsible for this work. I would like to thank you for the audience. I will give back to the moderator now.
Thank you so much, Ruth. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second presenter, Kevin L. Williams, Senior Scientist Endotoxin at BIOMÉRIEUX. Kevin studied microbiology at the Texas A&M University and has worked in the pharmaceutical field for 35 years. The bulk of his career was spent at Eli Lilly and Company for 30 years, developing QC tests for microbial and endotoxin detection. Kevin's current role at BIOMÉRIEUX as senior scientist focuses on endotoxin science, including low endotoxin recovery method development. Kevin also helped setting up a dedicated endotoxin service laboratory in the USA for hold time studies, LER root cause analysis, and endotoxin demasking protocol development. He is the author of Endotoxin second edition, Microbial Contamination Control in Parenteral Manufacturing, and Endotoxins third edition. Welcome, Kevin.
Thank you very much, and thanks to everyone for calling in today. Let me get on the right slide. So we're talking about low endotoxin recovery, specifically demasking, and High-Glos, which is a BIOMÉRIEUX company, has some unique product offerings in this area that we're going to discuss. Here's a general outline of the short talk today. I want to remind everybody why biologics are different from some historical drugs, small molecule drugs and large volume parenterals. And then we'll get into the mechanism of LER, talk a little bit about protein masking. It's not that different in the fact that what you put in as a spike can't be recovered. So we want to see, how do we know if we have masking and how that masking can be overcome. In biologics testing, early on it was realized that these drugs were going to be a bit more problematic. And some of the reason for this is that they're very large and complex molecules. If you think of a monoclonal antibody, it's about 150,000 Daltons. You can compare that to a small molecule cancer drug, such as gemcitabine, which is less than 400 Daltons. So those small molecules can enter into cells and make their effects known that way, whereas these large biologics bump around on the outside, and therefore they're under immune surveillance, and they interact with the cell surface signaling molecules as is shown here. Some of those cell surface signaling molecules are toll-like receptors, which of course detect endotoxin. So endotoxin can interact not only with the innate immune system that we're used to dealing with in endotoxin and fever responses, but also with the adaptive immune system. We've seen that in analytical testing in several different assays. This includes protein aggregation. There's been some immunogenicity from aggregated proteins, so now that's a test that's required to ensure that these aggregates aren't there. Siliconization, those have to be prevented as they occur in the stoppers, etc. Subvisible particles was a similar kind of paradigm change. Before biologics, they were only concerned about visible particles. And also the storage conditions have become important because they can contribute to these kind of changes. So in the end, pyrogenicity is not the only action of endotoxin. An example of this is in vaccinology. You can look up adjuvants, and endotoxin that's been detoxified, that's no longer fever-causing, is used as an immune stimulant in vaccines. So we can see there's a lot going on in terms of endotoxin with the adaptive immune system that we're not quite used to yet. So what is LER? We can go back to Dr. Chen's original presentation in 2013 at the PDA. This is a picture of the slide that he showed. We're used to seeing this slide now in terms of hold time recovery, where a spike into a direct spike into product diminishes over time and cannot be recovered. So we call LER the time-dependent inability to recover endotoxin from biologics formulations with LAL tests or recombinant factor C tests. This is not the first time that masking was shown. A PET showed 20 years ago that protein masking is prevalent with recombinant proteins. I'm going to talk a little bit about that as well. As far as the mechanism, this is from a Schwarz paper recently on masking, and you can see on the left-hand side that the most active form is thought to be the micelle, of endotoxins. So these micelle aggregates can be disaggregated by chelators, and the chelators have included citrate, EDTA, as Ruth mentioned, phosphate buffer, and then as a second step, a surfactant. Once the chelator is loosened up the micelle, the surfactant can wiggle in between the monomers and eventually dissociate them all, such as the aggregate can't be maintained, and this is the problematic part. If we have just citrate alone, that could be interfering, but it could be overcome by traditional methods, and also polysorbate alone can be overcome its effects if it's interfering with the LAL testing. But the combination of the two over time is really what has caused all the problems. Protein masking, I want to mention that quickly here. Protein masking, as shown as far back as 20 years ago when Pesh did this study, he took some both cationic proteins and anionic proteins. So you can see that the cationic proteins, which are positively charged, have a large propensity to bind endotoxin, which has a negative charge. So you can see at the top there, these three proteins bound what he used as a natural endotoxin. It was not very purified. It was grown up and just slightly purified. So we would call that an NOE today. And when he added it to the solutions, you can see the significant amount of binding. Got back less than 2% with human IgG, which of course is the scaffold for monoclonal antibodies. So from this data, you can see that protein binding is also a problem with biologics, but we've largely been able to not focus on this for the past years, but now it's going to come back into focus. These solutions didn't have any polysorbate or citrate. They also were relatively small concentrations at 1 milligram per mil, whereas today biologics are much more concentrated than that. So you can expect an exaggerated binding for some cationic proteins. I want to talk about the LER demasking platform that BIOMÉRIEUX has been using for some time now, based on a patent performed several years ago. There's 4 stages here to talk about, with the last one being conversion of the method to LAL. But first off, how do we know we have masking? Ruth had talked about this a little bit. We perform a direct spike hold time study. We perform this in the reverse manner, where the 7-day spike is made first, and then the 4-day, 2-day, 1-day, 4-hour, so on, such that all these can be put onto the same plate. Each of these spiking time points has a corresponding water control, just to verify that the spike was made appropriately. So at the end of this process, you end up with a 1-plate test intended to improve the variability rather than comparing several tests from several different runs. So we get a result that looks like this. This is typical LER, where you can see that after 24 hours, the initial spike has reduced to less than 50%, and then that reduction is maintained over the course of the hold. If it would have come back at the next time point after the initial low, that would have been acceptable, and the fact that it takes 2 in a row by definition to be considered LER. So after we've identified LER, how can we overcome this masking regardless of what the cause of the masking is, if it's LER, if it's protein binding. Either of those would show up in your hold time study, and we can address those kind of issues using EndoRS. It's a unique endotoxin recovery kit. This kit is shown here on the left. So it makes up some proprietary solutions that can be mixed and matched in order to do a screening test in order to find the best assay that will overcome many different kinds of formulations that have LER. This is one tool. Another tool is the EndoLISA. I'm going to talk a little bit about that, as well as these two tools can be used together to great effect to help to overcome the problem. So basically, the components of the EndoRS toolbox are designed to reverse the disaggregation. So in terms of starting from right to left here, we start with the masked LPS, so we'll mask some solutions of drug products for typically 7 days, and then we'll work with those solutions in a screening process to show which EndoRS methods best work to reverse the process. So we're taking the endotoxin that is there in the product, and we are disassociating it from the surfactant, which is covering the monomers now. And once we pulled that away, we can allow it to naturally reform the aggregates that are detectable in the LEL test. The components consist of what are labeled reagents A through E. A is a buffer. We want to start out with the same pH, always starting out from the same reference point. The so-called disturber, we want to destabilize the LPS surfactant complex because really the tenacity of that is what's keeping it, LPS from reaggregating. We can use the C, an absorber here, to take that surfactant away. And then the modulator, there's D1, D2. It supports this process. And finally, the so-called reconfigurator promotes the aggregate formation. We can see the most basic matrix can be performed here to discover the best starting point of the various demasking approaches, as shown on the left-hand side. So the most simple here is simply A and E. Depending on the formulation, this may be enough to overcome the demasking. In other cases, you'll want to include B, C, and or D1 or D2. So let's get into this in a little more depth here. Well, first we're going to look at EndoLISA as the second tool for demasking. And this is a plate kit, a test that actually has these little feet proteins from the bacteriophage that you see here in the top picture. These bacteriophage exist in nature, and they're highly specific to gram-negative bacteria. They bind here at the KDO, core sugar of LPS, to attach themselves to the bacteria. And it's the cloning and the cell culture production of these cells that allows for this assay. So the plates are coated with these proteins, and then the samples are incubated into wells. So this is a binding step, and then these solutions, typical of a ELISA test, which uses a monoclonal to grab structures in the solution. This uses these proteins, and then you can wash away the sample entirely, so you end up with an empty plate with a bound bound endotoxin from both the standards and the samples, and then you overlay this with recombinant factor C solution to do the detection. We can see the utility of this in a paper that was published in Nature showing the comparison of the ENDOLYSA tolerance for different substances. On the left-hand side, there's buffers, there's salt, chaotropic agents, organic solvents, detergents, chelators, protease inhibitors, antibiotics. So you can see in the comparison in the right-hand column of ENDOLYSA with LAL, you can see what the ENDOLYSA will tolerate. And let's take an example here, ethanol under the organic solvent. You can see a 30% tolerance in the ENDOLYSA versus a 0.5% tolerance in LAL. So that's about a 60% differential there, and likewise, these differentials can go up to 1,000-fold. So it's a good tool when you're looking at comparing different ENDO-RS treatments. So the demasking protocol development is a systematic procedure using a matrix approach for screening to see which of these mixing and matching of reagents is going to best support a test. And then once we have promising recoveries, we can optimize those by making further small iterations. Screening, optimization, evaluation, and finally lowering of the sensitivity. So let's go through an example. This is typical of the process. It's not the real numbers for confidentiality reasons, but it's numbers that are made to look like numbers that we've actually recovered. So initially, with the ENDOLYSA combined with the ENDO-RS, we'll have a number of screens to perform, and this is initially done with a rather high spike of 100 EUs per mil. So here we'll get a bunch of recoveries, some good, some bad, some ugly, so to speak, but invariably there will be some good recoveries amongst the matrix. So these are promising recoveries that we can further utilize to go downward in recovery. So if we'll start at 100, we will work our way down to 10 or 5, depending on what the specification is for the drug product. So we'll continue this iterative path until we reach much improved recoveries. Here's a next step, recovering down to 10 EUs per mil. You can see here these were spiked with 10. The recovery was 15, and then it says it's 187.5%. The reason that the math doesn't add up is because you have to account for the little amounts of ENDO-RS that were added to the dilution, so that increases the dilution a little bit. And once you've adjusted that, you can know the actual dilution and thus the actual recovery. So this demasking protocol development goes from high spike down to low spike, and then once you really have a low spike that's recovering well, we can convert that method over to LAL. LAL, we can use more extended dilutions based on the MVD, and then we can also utilize diluents other than LAL reagent water as necessary. So we have some tools there that extend the effort towards LAL. Once the assay is arrived at, we can transfer this to the customer lab, and the customer can validate this in their own laboratory as USP BET85, because in the end this is a sample treatment. If you end up adding A and E solutions to your drug test, then it's really no different than adding mag chloride or pyrisperse or some other treatments that are used or diluents that are used for BET85. So it shouldn't be seen as something that's not compendial in that regard. We've also set up a demasking service here in the Chicago area for U.S. customers, and we can look at the process. The process really is as I've already described. We'll look at the root cause for masking, analyze some of the components, start with the high spiking, work down to the low recovery, as low as the specification requires, and then work to transfer the method to the customer for a CGMP validated method. So the deliverables are a demasking protocol. You don't have to think of it as a demasking protocol. You can think of it as a routine QC test protocol, because in the end that's what it is. It's a recipe or a prescription for the test to be performed in a cookbook-like manner, just like any other LEL test. So also training and support packages can be put together, whatever the customer needs, and many customers, of course, can take the project on after an initial start to complete it themselves. And in the end of the demasking development protocol, we'll show in a hold time study additionally that this method has overcome the problems, and this will be part of the package that is delivered in terms of demasking. So I've gone through it pretty quickly. Maybe there's some questions. I do see one question here that I can answer real quick. Okay, I don't want to take all the questions. Go ahead.
Thank you, Kevin. If you would like to answer the questions, please do.
Okay, for the endotoxin hold time study, is it just the bulk drug substance tested? Well, I know this from the recent Lonza summit that Jessica Hankins from the FDA was there and gave a presentation, and she said, and I have the slides for these, that if you show no LER in your finished drug test, then you're basically finished. You don't have to show it for the bulk, because it's important to be able to test the finished drug. So that's an easy answer for that one. And the next one is also an easy answer. FDA is not accepting NOE spikes at this time. They're only accepting CSE or RSE. But it says, would you expect a similar outcome if an NOE was used? Well, the question's not very clear, because some NOEs are more subject to the phenomenon than others. We know that many of these isolates do demonstrate LER, but there are a few organisms that do not. So if you're studying the phenomenon, it wouldn't make any sense to use an organism that's not subject to the phenomenon. In terms of manufacturing control, you don't know what your contaminant's going to be, so you'll want to use something conservative, such as FDA says, CSE or RSE. So maybe Ruth wants to come on and answer a couple of questions.
Thank you so much, Kevin, for your presentation and for your answers. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that they can still send in their questions via the questions tab on the right-hand side of the screen. Was the MGS04 TRIS buffer successfully applied to solve all LER? Ruth?
Yes, we tried. So it is successful on 3 different clinical products, there it works. But I would like to emphasize that we not say that this buffer is the solution for all the issues that you can encounter. It is trial and error. You have to try, but this buffer works in our hands for 3 different clinical products.
Thank you, Ruth. Why do you think the LER issue has been so contentious? Kevin?
I think it's a new paradigm. It's definitely biologics are different than the previous drugs that we're all have so many years of experience with. So any paradigm change is usually painful. People say, well, this kind of testing doesn't make sense, and it doesn't make sense in the old context, but it certainly makes sense in the newer context when you look at some of the other changes that other analytical tests have gone through.
Thank you. Did you also collaborate with HighGloss to solve LER for your sample? Ruth?
Yes.
Hello?
I think I'm going to ask the question again. Did you also collaborate with HighGloss to solve LER for your sample? This is a question for Ruth.
Okay. Apparently you lost me. Hello. Can you hear me?
Yes.
We can hear you, Ruth.
Okay. So yes, our first clinical product. For that product, we collaborated indeed with HighGloss, and they developed for us a method that is useful as a solution for low endotoxin recovery. And then it is we were stressed by our management to develop our in-house method for that same product.
Thank you, Ruth. We don't have bioburden. How could we have contamination? Kevin?
I would refer you to the BPOG study on bioburden that was done in 2015, and they talk about how you expect to have some bioburden in these non-sterile processes that our biologics are produced under. They're not heat labile. They must be handled in a non-sterile manner. So along the process, you expect to have some bioburden. And this bioburden can be filtered out at these different process steps. But the filtering out of the organisms is not going to filter out the endotoxin or the other breakdown products of bacteria. So it's important to have the LAL assay or the RFC assay to supplement the bioburden studies. That BPOG paper from 2015 is a good one about that.
Thank you. Can you please clarify what you compare the endotoxin recovery for each time point to during a whole study? Is it compared to the T=0 recovery for the test material or to the theoretical spiked endotoxin at T=0? If this T=0 recovery in the test material, is this not an issue if inhibition is observed at T=0?
I can answer that question. It was on my in one of my slides, but maybe I did not mention it well enough. But that we have 3. Well, when we go back to the development slide and the acceptance criteria, there I have it is clearly stated that we work in our lab with 3 different acceptance criteria. First of all, the positive spike recovery, the PPC spike, that is your internal control, and that should be between 50 and 200%. Second, we have your water spike, that is your water that is spiked, that is your additional control, and that water spike is your time point 0. So for time point 0, your water spike is taken into account, and it should be between 50 and 200% of the spiked endotoxin. And then the third acceptance criteria is for the holding study. There we compare our time point zero of our product with the time point zero of the water. And that on time point zero should be between 50 and 200%. And for the other time points for the product, that should be between 50 and 200% of time point zero from the product. I hope that that is clear now.
Thank you, Ruth. Before I finish the webinar, I would like to ask our presenters, Ruth and Kevin, if they have any closing remarks. Ruth?
My closing remark is that this is a very, very, very challenging world now with the issues of low endotoxin recovery. But during the last years, there was a lot of debate and a lot of work ongoing to solve that issue and to help the FDA with the questions they came with. But I think after all that we are now in a good shape with that we see that there are solutions for this issue, and that when you can't handle it in your own hands, that there are also solutions and services available that you can take into consideration.
Thank you so much, Ruth. Kevin, do you have any closing remarks?
I'd just like to thank everyone for calling in, and if you have a question or a comment, you can certainly email me directly at kevin.williams@biomérieux.com. So I would love to speak with anyone about the topic. Thank you.
Thank you. Again, I would like to take this opportunity to thank our presenters, for Dr. Ruth Daniels, Senior Scientist, Microbiology Expert at Janssen, and Kevin L. Williams, Senior Scientist, Endotoxin at BIOMÉRIEUX, for sharing their knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone. Have a great day.